The understanding of the plant kingdom has evolved significantly over time, leading to a more nuanced classification of living organisms. Historically, organisms such as fungi, members of Monera, and Protista were grouped under the Plantae kingdom. However, advancements in biological research and genetic studies have resulted in the exclusion of these groups from Plantae due to their distinct characteristics and evolutionary pathways. For example, cyanobacteria, commonly referred to as blue-green algae, have been reclassified and are no longer considered true algae, illustrating the dynamic nature of scientific classification. In the early days of taxonomy, classification systems were simplistic and relied heavily on superficial morphological traits. These early systems focused on observable characteristics such as habit, color, and the number and shape of leaves. Such classifications, primarily based on vegetative traits or the structure of the androaceum, were often artificial. They failed to recognize the close relationships among species, leading to separations that did not accurately reflect evolutionary connections. The reliance on a limited set of characteristics meant that many closely related species were incorrectly categorized, obscuring the true nature of their relationships. As scientific understanding progressed, natural classification systems began to emerge. These systems were developed to reflect the natural affinities among organisms, taking into account not only external features, but also internal characteristics such as ultrastructure, anatomy, embryology, and phytochemistry. Pioneers like George Bentham and Joseph Dalton Hooker significantly contributed to this approach, providing a more holistic view of classification that recognized the complexity of life forms. Today, phylogenetic classification systems are widely accepted in the scientific community. These systems are based on evolutionary relationships, positing that organisms belonging to the same taxa share a common ancestor. This evolutionary perspective allows for a more accurate representation of the relationships among different organisms. Modern classification incorporates a variety of information sources, which is particularly important in cases where fossil evidence is lacking. One of the significant advancements in taxonomy is the development of numerical taxonomy. This method, facilitated by computer technology, analyzes all observable characteristics of organisms. Each trait is assigned a number or code, allowing for the processing of vast amounts of data. This approach ensures that each characteristic is given equal importance, enabling taxonomists to consider hundreds of traits simultaneously. The use of numerical taxonomy has revolutionized the field, providing a more objective basis for classification. In addition to numerical taxonomy, other specialized fields have emerged to aid in resolving classification challenges. Cytotaxonomy focuses on cytological information, such as chromosome number, structure, and behavior. This branch of taxonomy provides insights into the genetic makeup of organisms, helping to clarify their relationships. Chemotaxonomy, on the other hand, examines the chemical constituents of plants, utilizing biochemical data to address classification ambiguities. Together, these approaches enhance the accuracy and depth of taxonomic studies. Algae, a diverse group of chlorophyll-bearing organisms, play a vital role in ecosystems. They are primarily aquatic, thriving in both freshwater and marine environments, but can also be found in various other habitats, including moist stones, soils, and wood. Algae exhibit a wide range of forms and sizes, from colonial structures like volvox to filamentous forms such as Eulothrix and Spirogyra. Some marine algae, like kelps, can form massive plant bodies, showcasing the incredible diversity within this group. Reproduction in algae occurs through various methods, including vegetative, asexual, and sexual reproduction. Vegetative reproduction often involves fragmentation, where each fragment develops into a new thallus. Asexual reproduction typically produces different types of spores, with zoospores being the most common. These flagellated, motile spores germinate to give rise to new plants. Sexual reproduction in algae can be quite complex, involving the fusion of gametes that can be flagellated and similar in size, known as isogamous, or dissimilar in size, termed anisogamous. Oogamous reproduction, seen in species like Volvox, involves the fusion of a large, non-motile female gamete with a smaller, motile male gamete. Algae are not only ecologically significant, but also beneficial to humans in numerous ways. They are responsible for a substantial portion of carbon dioxide fixation on Earth through photosynthesis, contributing to the oxygen levels in aquatic environments. As primary producers, algae form the foundation of food webs, supporting a diverse range of aquatic life. Many species, such as Porphyra, Laminaria, and Sargassum, are consumed as food, while others yield commercially valuable substances. For instance, certain marine brown and red algae produce hydrocolloids like algin and carrageenan, which are widely used in the food industry. Chlorella, a unicellular alga rich in proteins, has gained popularity as a food supplement, even among astronauts. Algae are classified into three main groups: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae, commonly known as green algae, can be unicellular, colonial, or filamentous. They are typically green, due to the dominance of chlorophyll A and B, with pigments localized in distinct chloroplasts. These chloroplasts can vary in shape, and many species contain pyrenoids for starch storage. Vegetative reproduction in green algae occurs through fragmentation or spore formation, while asexual reproduction involves flagellated zoospores. Sexual reproduction shows considerable variation, with types including isogamous, anisogamous, and oogamous. Common green algae include Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Chara. Phaeophyceae, commonly referred to as brown algae, are predominantly found in marine environments and exhibit a remarkable diversity in size and form. These organisms range from simple branched filamentous types, such as Ectocarpus, to the more complex and profusely branched kelps, which can attain heights of up to 100 meters. This variation allows brown algae to adapt to different ecological niches within the ocean. Brown algae are characterized by their unique pigment composition, which includes chlorophyll A and C, carotenoids, and xanthophylls. These pigments contribute to their coloration, which can vary from olive green to various shades of brown. The specific color is often influenced by the presence of the xanthophyll pigment fucoxanthin, which plays a significant role in photosynthesis by absorbing light energy. In terms of energy storage, brown algae primarily store food as complex carbohydrates, notably laminarin or mannitol. The vegetative cells of brown algae possess a cellulosic wall, typically coated on the outside with a gelatinous substance known as algin. This algin serves multiple purposes, including protection and moisture retention in their marine habitats. The structure of brown algae usually includes a holdfast that anchors the plant to the substrate, a stipe that functions as a stalk, and a frond that serves as the leaf-like photosynthetic organ. Reproductive strategies in brown algae are diverse. Vegetative reproduction often occurs through fragmentation, where parts of the algae break off and develop into new individuals. Asexual reproduction is primarily facilitated by the production of biflagellate zoospores, which are pear-shaped and equipped with two unequal laterally attached flagella. This adaptation allows the zoospores to swim through the water, increasing their chances of finding suitable environments for settlement. Sexual reproduction in brown algae can be categorized as isogamous, anisogamous, or oogamous, depending on the species. In isogamous species, the gametes are similar in morphology, while in anisogamous species, they differ in size. Oogamous species produce larger, non-motile eggs and smaller, motile sperm. The fusion of gametes can occur in the water or within the oogonium of the female. The gametes are also pear-shaped and flagellated, facilitating their movement toward each other for fertilization. Common examples of brown algae include Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus, each playing a vital role in their respective marine ecosystems. Rhodophyceae, or red algae, are another significant group of algae recognized for the predominance of the red pigment R-phycoerythrin. Most red algae are marine, with higher concentrations found in warmer waters. They thrive in both well-lit areas near the water's surface and at considerable depths in the ocean, where light penetration is limited. This adaptability allows red algae to occupy a wide range of habitats. The red thalli of red algae are typically multicellular and store food as Floridian starch, which is structurally similar to amylopectin and glycogen. This carbohydrate storage is essential for their energy needs. Red algae generally reproduce vegetatively through fragmentation, enabling them to spread and colonize new areas. Asexual reproduction occurs via non-motile spores, while sexual reproduction involves non-motile gametes. The sexual process is oogamous and is accompanied by complex post-fertilization developments. Notable members of the Rhodophyceae include Polysiphonia, Porphyra, Gracilaria, and Gelidium, each contributing to the ecological balance of their environments. Bryophytes, which encompass various mosses and liverworts, are commonly found in moist, shaded areas, particularly in hilly regions. They are often referred to as the amphibians of the plant kingdom, due to their ability to live in soil while relying on water for sexual reproduction. Bryophytes typically thrive in damp, humid, and shaded localities, making them essential for maintaining moisture in their ecosystems. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and can be either prostrate or erect, anchored to the substrate by unicellular or multicellular rhizoids. Bryophytes lack true roots, stems, or leaves, although they may possess structures that resemble roots, leaves, or stems. The main plant body is haploid and functions as a gametophyte, producing gametes. The male sex organ, called antheridium, produces biflagellate antherozoids, while the female organ, known as the archegonium, is flask-shaped and produces a single egg. Fertilization occurs in water, where an antherozoid swims to the archegonium to fuse with the egg, forming a zygote. This zygote does not undergo immediate reduction division. Instead, it develops into a multicellular body called a sporophyte. The sporophyte remains attached to the photosynthetic gametophyte, deriving nourishment from it. Some cells of the sporophyte undergo meiosis to produce haploid spores, which germinate to form new gametophytes. While bryophytes are generally of limited economic importance, certain mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a type of moss, are particularly valuable for their water retention capabilities and have been utilized as fuel and packing material for transporting living materials. Mosses, along with lichens, are often the first organisms to colonize rocks, playing a crucial role in soil formation. They decompose rocks, creating a substrate suitable for the growth of higher plants. Additionally, mosses form dense mats on the soil, which help reduce the impact of falling rain and prevent soil erosion. Liverworts typically thrive in moist, shady habitats such as stream banks, marshy ground, damp soil, and tree bark. The plant body of liverworts is thalloid, exemplified by Marchantia. Their thalli are dorsiventral and closely oppressed to the substrate. Asexual reproduction occurs through thallus fragmentation, or the formation of specialized structures called gemmi, which are green, multicellular, asexual buds that develop in small receptacles known as gemma cups. These gemma detach from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs may be produced on the same or different thalli. The sporophyte is differentiated into a foot, seta, and capsule, where meiosis occurs to produce spores that germinate into free-living gametophytes. Rhodophyceae, commonly known as red algae, are fascinating organisms that thrive primarily in marine environments. Their vibrant red coloration is primarily due to the presence of R-phycoerythrin, a pigment that allows them to absorb light efficiently in deeper waters. Most red algae are found in warmer regions, where they flourish in both well-lit areas near the surface and in the depths of the ocean, where light penetration is minimal. This adaptability to varying light conditions is one of the many remarkable features of Rhodophyceae. The structure of red algae is predominantly multicellular, with some species exhibiting complex body organization. Unlike many other algae, red algae store their food in the form of Floridean starch, which shares structural similarities with amylopectin and glycogen. This unique storage method allows them to thrive in diverse environments and provides essential energy reserves. Reproduction in Rhodophyceae is equally intriguing. They typically reproduce vegetatively through fragmentation, where pieces of the algae break off and grow into new individuals. Asexual reproduction occurs via non-motile spores, while sexual reproduction involves non-motile gametes. The sexual process is oogamous, meaning it involves the fusion of a larger female gamete with a smaller male gamete. This process is accompanied by complex post-fertilization developments, leading to the formation of new individuals. Notable members of this group include Polysiphonia, Porphyra, Gracilaria, and Gelidium, each contributing to the rich biodiversity of marine ecosystems. Transitioning to bryophytes, this group includes various mosses and liverworts that commonly inhabit moist, shaded areas, particularly in hilly regions. Bryophytes are often referred to as the amphibians of the plant kingdom due to their unique ability to live on land while still relying on water for sexual reproduction. These plants typically thrive in damp, humid, and shaded localities, playing a crucial role in plant succession on bare rocks and soil. The plant body of bryophytes is more differentiated than that of algae, featuring a thallus-like structure that can be either prostrate or erect. They attach to the substrate using unicellular or multicellular rhizoids, which serve a similar purpose to roots but lack true root structures. Bryophytes may possess root-like, leaf-like, or stem-like structures, but their main plant body is haploid and is referred to as a gametophyte. This gametophyte produces gametes, with male and female sex organs being multicellular. In bryophytes, the male sex organ is called antheridium, which produces biflagellate antherozoids. The female sex organ, known as archegonium, is flask-shaped and produces a single egg. The antherozoids are released into water, where they swim to the archegonium, leading to fertilization. After fertilization, the zygote does not undergo immediate reduction division. Instead, it develops into a multicellular body known as a sporophyte. This sporophyte is not free-living. It remains attached to the photosynthetic gametophyte, deriving nourishment from it. Some cells of the sporophyte undergo reduction division, meiosis, to produce haploid spores, which germinate to form new gametophytes. While bryophytes may not hold significant economic importance, they play vital ecological roles. Some mosses provide food for herbaceous mammals, birds, and other animals. Species like sphagnum, a type of moss, are particularly noteworthy as they produce peat, which has been used for fuel and as packing material due to its water retention capabilities. Mosses, along with lichens, are often the first organisms to colonize rocks, contributing to soil formation and making the substrate suitable for higher plant growth. Their dense mats on soil help reduce the impact of falling rain, preventing soil erosion and promoting a healthier ecosystem. Delving deeper into bryophytes, liverworts are a prominent subgroup that typically grow in moist, shady habitats such as stream banks, marshy grounds, damp soils, and tree bark. The plant body of liverworts, such as Marchantia, is thalloid, meaning it has a flat, ribbon-like structure that closely adheres to the substrate. Leafy liverworts feature tiny leaf-like appendages arranged in 2 rows along stem-like structures. Asexual reproduction in liverworts occurs through thallus fragmentation, or the formation of specialized structures called gemmae. Gemmae are green, multicellular asexual buds that develop in small receptacles known as gemma cups located on the thalli. When gemmae detach from the parent body, they germinate to form new individuals. During sexual reproduction, male and female sex organs can be produced on the same or different thalli. The sporophyte in liverworts is differentiated into a foot, seta, and capsule, where meiosis occurs to produce spores that germinate into free-living gametophytes. Mosses, another significant group of bryophytes, predominantly exhibit a gametophyte stage in their life cycle. This stage consists of 2 phases: the protonema stage, which develops directly from a spore, and the leafy stage, which arises from the secondary protonema as a lateral bud. The leafy stage consists of upright, slender axes bearing spirally arranged leaves attached to the soil through multicellular, branched rhizoids. This stage also bears the sex organs. Vegetative reproduction in mosses occurs through fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs, antheridia and archegonia, are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte, which is more elaborate than that in liverworts. The sporophyte consists of a foot, seta, and capsule, where spores are formed following meiosis. Mosses have developed intricate mechanisms for spore dispersal, ensuring their survival and propagation in various environments. Common examples of mosses include Funaria, Polytrichum, and Sphagnum, each playing essential roles in their ecosystems and contributing to the overall biodiversity of plant life. Pteridophytes, a fascinating group of plants, include horsetails and ferns and are known for their unique characteristics and ecological importance. These plants are not just visually appealing, they also serve various practical purposes, including medicinal uses and soil binding capabilities. Pteridophytes thrive in cool, damp, and shady environments, although some species can adapt to sandy soils, showcasing their resilience and versatility. Evolutionarily, pteridophytes hold a significant position as the first terrestrial plants to develop vascular tissues, specifically xylem and phloem. This advancement allowed them to transport water and nutrients efficiently, enabling them to grow larger and occupy diverse habitats. The main plant body of pteridophytes is the sporophyte or shorebite, which is differentiated into true roots, stems, and leaves. These organs possess well-differentiated vascular tissues, marking a critical step in plant evolution. The leaves of pteridophytes vary significantly in size and shape. Some species, like Selaginella, have small leaves known as microphyls, while others, such as ferns, feature large leaves called macrophyls. This diversity in leaf morphology allows pteridophytes to adapt to different environmental conditions and optimize their photosynthetic efficiency. Sporophytes produce sporangia, which are structures that contain spores. These sporangia are often subtended by leaf-like appendages called sporophylls. In certain cases, sporophylls can form distinct compact structures known as strobili or cones, as seen in genera like Equisetum and Selaginella. The sporangia undergo meiosis to produce spores, which are then released into the environment. Once released, the spores germinate to give rise to small, inconspicuous, multicellular, free-living gametophytes called prothallus. These gametophytes are primarily photosynthetic and require cool, damp, and shady places to thrive. The specific environmental requirements for gametophyte growth, along with the necessity for water during fertilization, limit the geographical distribution of living pteridophytes. The gametophytes bear male and female reproductive organs known as antheridia and archegonia, respectively. Water plays a crucial role in the fertilization process, as it facilitates the transfer of antherozoids, the male gametes released from the antheridia, to the mouth of the archegonium. The fusion of the male gamete with the egg within the archegonium results in the formation of a zygote, which subsequently develops into a multicellular, well-differentiated sporophyte. This sporophyte represents the dominant phase of the pteridophyte life cycle. In the majority of pteridophytes, all spores produced are of similar kinds, classifying them as homosporous. However, some genera, such as Selaginella and Salvinia, produce two distinct types of spores: macro, large, and micro, small, spores, making them heterosporous. The megaspores germinate to form female gametophytes, while microspores develop into male gametophytes. Interestingly, in heterosporous pteridophytes, the female gametophytes are often retained on the parent sporophytes for varying periods, allowing the development of zygotes into young embryos within the protective environment of the female gametophyte. This process is considered a precursor to the evolution of seeds, marking a significant advancement in plant reproduction. Pteridophytes are further classified into four primary classes: Xylopsida, which includes the whisk ferns; Lycopsida, encompassing club mosses like Selaginella and Lycopodium; Sphenoptida, represented by horsetails like Equisetum; and Pteropsida, which includes true ferns such as Dryopteris, Pteris, and Adiantum. Each class exhibits unique characteristics and adaptations, contributing to the overall diversity of this ancient plant group. Transitioning to gymnosperms, these plants are characterized by their naked seeds, meaning their ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. Gymnosperms encompass a variety of forms, including medium-sized trees, tall trees, and shrubs. One of the most notable gymnosperms is the giant redwood tree, Sequoia, recognized as one of the tallest tree species on Earth. The roots of gymnosperms typically consist of taproots, although some genera, like Pinus, form associations with fungi in the form of mycorrhiza, while others, such as Cycas, develop specialized roots called coralloid roots that associate with nitrogen-fixing cyanobacteria. The stems of gymnosperms can be unbranched, as seen in Cycas, or branched, as in Pinus and Cedrus. Their leaves may be simple or compound, with Cycas exhibiting pinnate leaves that persist for several years. Gymnosperm leaves are well adapted to withstand extreme temperatures, humidity, and wind. In conifers, needle-like leaves reduce surface area, while thick cuticles and sunken stomata help minimize water loss, making them highly efficient in arid environments. Gymnosperms are also heterosporous, producing haploid microspores and megaspores within sporangia borne on sporophylls arranged spirally along an axis to form strobili or cones. Male strobili, containing microsporophylls and microsporangia, produce pollen grains which represent a highly reduced male gametophyte. Conversely, female strobili bear megasporophylls with ovules. The male and female gametophytes in gymnosperms do not exist independently. They remain within the sporangia on the sporophytes. Pollen grains are released from microsporangia and carried by air currents to the ovules on megasporophylls. The pollen tube grows toward the archegonia in the ovules, discharging male gametes near the archegonia's mouth. Following fertilization, the zygote develops into an embryo, while the ovules transform into seeds, which remain uncovered. Understanding the evolutionary significance of pteridophytes and gymnosperms reveals the complexity of plant life on Earth. These ancient plants not only contribute to biodiversity, but also play vital roles in ecosystems and human applications, highlighting their importance in both natural and cultivated environments. Angiosperms, or flowering plants, represent a remarkable group of plants that thrive in diverse habitats. Unlike gymnosperms, where ovules remain exposed, angiosperms develop their pollen grains and ovules within specialized structures known as flowers. This unique feature not only aids in reproduction, but also enhances their adaptability. The seeds of angiosperms are encased in fruits, providing protection and facilitating dispersal. This adaptation allows angiosperms to occupy a wide range of environments, from wetlands to arid deserts. They exhibit incredible diversity, with sizes ranging from the minute wolffia, the smallest flowering plant, to towering eucalyptus trees that can exceed 100 meters in height. Angiosperms play a crucial role in human life and the ecosystem. They are a primary source of food, supplying fruits, vegetables, grains, and nuts that nourish billions worldwide. Additionally, they provide fodder for livestock, fuel for energy, and raw materials for countless products, including medicines and textiles. This vast group of plants is classified into two main categories: dicotyledons and monocotyledons. Dicotyledons, characterized by their two seed leaves, include familiar plants like roses and sunflowers. Monocotyledons, with a single seed leaf, encompass grasses, lilies, and orchids. The ecological importance of angiosperms cannot be overstated. They contribute to soil health, support wildlife, and play a vital role in carbon sequestration. As the most diverse group of plants on Earth, angiosperms continue to evolve, adapting to changing environments and human needs. Their significance in agriculture, horticulture, and medicine highlights the need for conservation efforts to protect these essential plants for future generations.